Hello Year 10 and welcome to this second recorded lesson on the reproductive topic, B13.2. This lesson is entitled Cell Division in Sexual Reproduction and can be found on pages 198 and 199 in your biology textbook. So in this lesson we're going to try and identify how gametes form, describe what happens during meiosis, and compare mitosis and meiosis and explain why gametes are genetically different. So before we begin on meiosis, which is the process involved in gamete formation and sexual reproduction, we're going to have a quick recap on mitosis, which you learned about in topic B2. So if you remember, mitosis is about the doubling of the DNA in the nucleus, followed by the dividing of that DNA into two identical halves and the completion of new nuclei and new cell membranes so that we have two identical cells. And this is used for reproduction of some organisms, asexual reproduction, and then for growth and repair in all organisms. So here is the process in more detail. This takes us up towards A level, and at GCSE you will not need to be able to quote each of the names that are on here, but there's no harm in you having that as background knowledge. So when we join the process of mitosis, the DNA has already been doubled, it has already been copied, and that's why the chromosomes are represented as these double strands here. And the reason they're represented in different colours is because originally one of each pair of chromosomes will have come from the mother and one will have come from the father. And we therefore can recognise that this is one pair of chromosomes of the 23 pairs that are found in humans. So in mitosis, those chromosomes do not behave as pairs. They all line up along the equator. So you would have all 46 chromosomes that have been doubled lining up along the equator like this in metaphase. In anaphase, something called spindle fibres, that's these grey lines here, form, and they attach to the middles of the pairs of doubled chromosomes and they pull them apart. So what that means is you have got a single set of chromosomes in each side. So this would be 46 chromosomes here, 46 chromosomes here. And then the new nuclei can form around each set of chromosomes. New cell membranes can form around each nucleus, and some of the organelles, the ribosomes and mitochondria, will be in each of these cells. And this process is called cytokinesis, which quite literally means cell splitting. So we have ended up with two identical cells from the one original one. So just to make sure that that makes sense to you, here are a sequence of descriptions. You need to pause the video here and to have a go at putting these phrases in the correct order. So this is the order you should have come up with. Each chromosome has already been copied and now consists of two identical strands. The double-stranded chromosomes line up at the equator of the cell. Spindle fibres pull the doubled chromosomes apart. A new nucleus forms around each set of chromosomes. The cell splits to include one nucleus in each daughter cell, and the two daughter cells are identical. So this is mitosis, remember. So let's move on now to consider meiosis. So mitosis is involved in asexual reproduction, whereas meiosis is involved in sexual reproduction. In sexual reproduction, there is always the formation of gametes, and these gametes have only half the original number of chromosomes. So when the gametes, the egg and the sperm, or the ovule and the pollen, get back together during fertilization and join or fuse together, you increase the number of chromosomes back up to the original number. So in humans, that will be from 46 chromosomes into 23 chromosomes in each egg and in each sperm, and then in a fertilized egg or a zygote, we will be back up to 46 chromosomes. So here we begin to compare the two processes. In mitosis, you can see that we are starting with 4 chromosomes in this model. Those chromosomes are doubled and have the 2 strands to them, and then pulled apart so that we get 1 set of chromosomes in each of the 2 cells. 4 chromosomes in each cell, same as with the parent, and the 2 cells that have been produced are identical to each other. In meiosis, the doubling of the chromosomes happens in the same way. However, rather than all the chromosomes lining up along the equator to be pulled apart into their identical pairs, the first division that takes place are the pairs of doubled chromosomes. So both chromosome 1 will pair up, both chromosome 2 will pair up, and in humans all the way up to chromosome 23 will pair up. And those 2 pairs are pulled apart from each other. There is then a second division, which is more similar to mitosis, whereby the doubled chromosomes line up along the equator and are pulled apart. And this results in only half the number of chromosomes in each of the 4 daughter cells. There's lots of writing on the next 2 slides, so at any point if you want to stop the commentary and read what's written there, then that will help you with your understanding as well. So in a normal human cell, we will have 23 paired chromosomes. And the thing to remember, as I mentioned before, is that originally one of those chromosomes will have come from the mother of this person, and one of these chromosomes will have come from the father. The other thing to remember is that each chromosome 1 will hold a gene for a particular characteristic. So let's say, for example, the gene for eye colour is on chromosome 1. It means that every human will have a gene for eye colour on each of its chromosome 1s, meaning that you have 2 copies of a gene for each characteristic. We'll learn more about how it is decided upon which of those characteristics gets passed on in a future lesson. The important thing to note is that because these chromosomes have come from different parents, the allele, as we call it, the copy of the gene for eye colour on one chromosome may be different to the copy of the allele of the gene for the other. So for example, this chromosome may carry the blue-eyed gene, this chromosome may carry the brown-eyed gene. They have come from different parents. Now that is true of all of the chromosomes in the human genome. And the other thing to note is that we don't know whether the chromosome on the left was the maternal one and the one on the right was the paternal one, and whether that's different in each of the pairs. So when the chromosomes line up for their divisions, the mixture of chromosomes which are passed into each egg and each sperm are going to be different. And that is what makes meiosis create variation for the next generation. The other thing to note from this picture is that when the chromosomes double, remember they stay joined, and in the first, or in the mitotic division, it's these doubled joined chromosomes which are being separated back into their individual strands like this. Whereas in meiosis, the first division that takes place is the separation of these doubled pairs. So one chromosome 1, which has been doubled, goes into one part of the cell and the other goes into the other part of the cell. In the second division, each doubled strand is separated. This includes the sex-determining chromosomes. This organism here is a male because he has got an X and a Y chromosome in his genome. However, when the gametes are produced, Either an X chromosome or a Y chromosome will go into each sperm. This means that when that sperm fertilizes an egg, it will either add another X chromosome to the egg or it will add a Y chromosome to the egg. All eggs have only X chromosomes and all females have only X chromosomes. Therefore, the egg always carries an X. So if a sperm carrying an X joins it, the child will be female. If a sperm carrying a Y joins it, it will be male. So the key thing about this assortment of the genes, the moving around, the pairing up, the dividing, the separating into different cells, the production of these eggs and sperm, all of this is about mixing and matching these chromosomes. There's one more feature, which is that when these chromosomes are lined up like this, they are actually able to swap a little bit of their DNA. So a section of the chromosome here might swap with its opposite section here. And that's called crossing over. And that further increases the variation so that the chromosomes being passed into each egg and each sperm will be a random assortment of maternal and paternal chromosomes from each parent, and that there will have been some crossing over taking place, which further mixes the genes available. And then if you think about the idea that the egg and the sperm that eventually end up as fertilizing each other, then the variation is increased again, because millions of sperm are produced in each ejaculation and the female holds thousands of eggs in her ovary and just one will develop each month to be released, and therefore the particular combination of genes that end up in each egg and each sperm in each successful fertilization is very random indeed, which is why you are all individuals. So again, I would like you to make sure you've understood the process of meiosis. I would like you to pause the video and put these phrases into order before checking the answers on the next slide. So here are our answers. Each chromosome has already been copied and now consists of 2 identical strands. The pairs of doubled chromosomes line up at the equator. Spindle fibers separate the pairs of chromosomes, each doubled chromosome being pulled to opposite ends of the cell. Excuse me. Doubled chromosomes now line up along the new equator of each cell. Spindle fibers pull the doubled chromosomes apart so that there are now 4 groups of individual chromosomes. Nuclei form around each group of single chromosomes, and each group of chromosomes is different to the other and will now develop into a gamete. Sperm and eggs in mammals and pollen and ovules in plants. So if you remember, we were going to aim to compare mitosis and meiosis. So a similarity would be that we start with 46 chromosomes in each of the cells. However, in mitosis, once the genetic material has been doubled and copied, it splits into 2 identical cells, each of which also have 46 chromosomes in humans. However, in meiosis, the doubled chromosomes split into 4, each of which has only 23 chromosomes, and each of those new cells is genetically different to each other. Mitosis is used in growth and repair and asexual reproduction. Meiosis is used only to produce gametes in sexual reproduction. When mitosis takes place, there is no variation and the new cells that are produced are identical. In meiosis, there is variation and the new cells that are produced are genetically different to each other. We can also use the words diploid and haploid. Diploid means that there is 2 copies of each chromosome. So here we're talking about 23 pairs of chromosomes, 46 in total, meaning the cell is diploid. These two cells are also diploid. This cell is diploid. It contains 23 pairs, 46 chromosomes altogether. These cells are haploid. They contain half the number of chromosomes and only a single copy of each numbered chromosome. When they join back up together, so if this was an egg cell, it would join with a sperm cell from another organism, from a male, and the 23 together would make the 23 pairs, or the 46, the diploid zygote. So one way that you could begin to put your ideas down on paper might be to use a Venn diagram. You could put the mitosis information in this part of the diagram. You could put the meiosis information in this part of the diagram and any similarities in the middle here. You are going to use this information to help you to answer a 6-mark question in a moment. So pause the video here and complete that task. There is also a stretch and challenge task on this slide, which says, imagine a world in which there is no meiosis. Imagine what effect that would have on us as organisms. So here is your 6-mark question. Meiosis and mitosis are different types of division in human cells. Compare the two processes by referring to where each takes place and the kind of products that are made. So you are going to have a think about how you would describe simply, because it's only 6 marks, what is happening in mitosis, what is happening in meiosis, what the similarities are, and what the differences are. Remember, in a 6-mark question, you are aiming to communicate a new point with each sentence that you make, and that you are aiming to stick to the facts. Okay? Good luck. I hope this has been useful. Obviously, you can go back through the video, listen again, read again, and make sure that you use the textbook pages 198 and 199 to support your learning. There are also some questions on there, so if you wanted to have a go at those first, by all means do so. Make your own notes as you have been, and keep working in a way that really works for you. Okay? Take care, and I'm sure I shall see some of you before long. Bye-bye.